I shortened the title from what you all received. I took out the hearing aid part because I spoke about that on Friday. So this is going to be about genetics, gene therapy, and cochlear implants, and in no particular order. And just as sort of an intro, I'm certainly not a geneticist and certainly not a gene therapy scientist. And in fact, for years while I was at Iowa, Richard Smith was always telling me I need to get genetics on all of my patients with hearing loss. And I kept saying, why? What difference is it going to make to what I do? And now it actually does make a difference. And increasingly, it will be making an even bigger difference, as you'll see in some of the things that I present. So for disclosures, the University of Washington is a trial site for the Regeneron gene therapy product. None of the gene therapies to be discussed are approved by the FDA outside clinical trials. I've previously received research funding from Decibel, which was subsequently acquired by Regeneron, for safety studies in non-human primates of their gene therapy product, as well as its surgical delivery. And I've been a consultant for Advanced Bionics and Hemidina, which is a company that was trying to develop a novel cochlear implant. This is the family tree of cochlear implants. And part of why it's really fun for me to be here is that even though I came here as a medical student, I think for 2 weeks. I spent 2 weeks here and 2 weeks at UCSF, which is when I first met Dr. Jackler, a very long time ago. And that's the only time I ever actually spent at Stanford. But Stanford played a really critical role in my choice of careers. And the reason why is partially illustrated in this figure, which I got from Don Eddington. Who is Don Eddington? Don Eddington invented the Interaid cochlear implant when he was a graduate student at Utah. Utah played another very important role early in cochlear implant history. The Interaid was the first multi-channel implant that was actually implanted in a large number of people. And if you go through the slide, the y-axis is history going back to Volta in 1790. And as you proceed inferiorly, you go forward in time. And this shows all of the research groups that were involved with cochlear implants in different eras, leaping forward to the 1950s with Giorno and Irie, and then the 1960s with House and Doyle, and initially Blair Simmons. And then moving into the 1970s when Blair recruited Bob White, who was the chair of electrical engineering at Stanford at the time, to initiate the Stanford cochlear implant project. All of these teams, then you progress forward and you see what devices they created, some of which ended up not going anywhere, some of which ended up being acquired by Cochlear, and some of which became the companies that we know now. So here's an image from Stanford's News Bureau in 1965. I thought when I was going to give this presentation that there was nobody in the room who was born yet besides maybe me in 1965, but obviously that's not the case. But I was 5 years old in 1965. And if you read the text, an experiment was being performed by Dr. F. Blair Simmons, assistant professor of surgery, and Dr. John Epley, a resident in otolaryngology at Stanford. It's extraordinary to see stuff like this from that era. But I said that Stanford played a key role in my choice of career, and this is why. So this is the Stanford Engineering Alumni magazine, circa 1980. And I was a junior in college, and I had won the lottery for an off-campus apartment. I moved in, and the previous resident of my apartment was a graduate student in engineering who'd been an undergraduate engineering student at Stanford. And so he received this magazine in the mail after he moved out and I moved in. So I picked it up and I said, gee, that looks really interesting. And at the time, I was just developing an interest in neuroscience, and I was working in a digital design lab. And I said, wow, and I was doing speech processing. We were trying to develop systems for speech recognition, which was a pipe dream at the time. I saw this and I read this article and I said, this is what I want to do. And it's really fun to be back here and actually talking about this now. Some of you may have seen this image. This is the first multi-channel programmable cochlear implant speech processor. And these are the 3 graduate students of Bob White's who built it. It was built on LSI technology, which was the cutting edge technology available at the time, and it ran on D-cell batteries. There was an exhibit on it in the Computer Museum for a little bit a while ago. And Les Atlas ended up moving to the University of Washington after he graduated with his PhD from Stanford, and that's where I met him. And he was on my dissertation committee when I did my PhD, and we've been colleagues since. He's retired now. Part of Stanford's cochlear implant legacy is the CIAP meeting. The first CIAP meeting was in 1983. It was a Gordon conference, and it was Bob White wrote the proposal to the Gordon folks to allow it to happen, and Blair Simmons is sitting over here. And basically everybody in the world, except for a couple of Australians who worked on cochlear implants at the time, were at this meeting. This meeting still goes on today, and at the end, I'm going to try and convince you to come to it because the next one is in Lake Tahoe in July. For those of you who don't know who the vice chair of the meeting was Dennis Klatt. And Dennis invented the Klatt synthesizer, which was the first speech-to-text synthesizer. And you actually know what Klatt's voice sounds like because the voice of Stephen Hawking is Dennis Klatt's voice. Because when they first created the technology, they used his voice as the model. And a few years later, they figured out how to use samples of somebody's voice back when they could talk and change it to that. And Dennis apparently went to Stephen Hawking and said, hey, would you like to have your own voice back? And Hawking said, nah, I like your voice. So I'm going to breeze through this and focus on the fact that we are now in the gene therapy era of cochlear implants. And what do I mean by that? What I mean is we're now going to be dealing with patients who may have had gene therapy in one ear and a cochlear implant in the other ear. They may have had failed gene therapy in one ear followed by a cochlear implant. There's questions as to whether anybody's going to end up getting gene therapy after they've had a cochlear implant. These 2 technologies are now merged, and I consider this the beginning of a new era in cochlear implantation. So let's do a little background. This is a study done by one of our students, Ryan Carlson, currently applying for residency in otolaryngology. And he did his PhD with Mary Claire King, who's a world-renowned geneticist. I hooked him up with our patient population. And he was able to identify a genetic cause of hearing loss in about half of over 400 pediatric patients. Unsurprisingly, GJB2 was the most common cause of hearing loss. And the second most common, and that was 30% of the genetic hearing losses, or 15% of all pediatric hearing loss in our population. The second most common one was STRC, which is what I spoke about on Friday, and relates to hearing aids, because STRC causes a moderate hearing loss with normal speech discrimination and quiet. Not patients that currently receive cochlear implants, and not patients who are likely to receive gene therapy anytime soon, although I'm going to describe some experiments that we need to do to get to the point where you might be able to do gene therapy in an ear with STRC. SLC26A4 was the 3rd most common large vestibular aqueduct. And if you go down the list, otoferlin was 1% of our population. As I implied, these percentages depend on sources of patients. I had never heard of STRC before this study because all the series I looked at were all series that came out of cochlear implant programs. and they're not going to have any STRC patients. And so it's always, in those groups, it's always GJB2 first and SLC26A4 second. A number of folks in this department are really interested in LOX-HD1. And I had to go back and look, and it's it's comparably common to otoferlin, actually, and another gene that's going to end up being of interest in the near future in terms of gene therapy. If you go to a different place than Seattle, and you go to Usami's lab in Japan, he's got huge data sets of patients, and in his case, 40% of 10,000 patients of all ages received a genetic diagnosis. And in his population, again, GJB2 was first, cadherin 23 was second, SLC26A4 was third, and STRC was fourth. But if you look at the percentages, it's actually roughly similar to what we see in the United States. And interestingly, only one genetic diagnosis in 10,000 patients was unilateral. So chasing down the genetics of unilateral hearing loss is going to be not something I would spend a lot of time on. So there are currently 5 clinical trials of gene therapy for deafness going on, run by 5 different companies, 2 of them in China, one in Shanghai and one in Nanjing, one in Montpellier, and 2 from Boston. Akuos was which was founded by Mike McKenna, was acquired by Eli Lilly, and Decibel, which was founded by other folks from Mass Ioneer, was acquired by Regeneron. And all 5 of these companies are chasing the same rare gene, otoferlin. And why are they doing that? Sort of worldwide, with the exception of Spain, otoferlin is anywhere from 1% to 2% of genetic deafness. So it's not terribly common, but not profoundly rare either. Spain is the outlier. About 8% of genetic deafness in Spain is otoferlin based. So Spain is obviously a site for clinical trials for this. The gene is too large for a single AAV vector, so that's a complication. So an AAV vector can only carry about 4,800 base pairs. So any gene that's bigger than 4,800 base pairs, you have a problem. And but the good news is that dual AAV therapy has been demonstrated to be effective in mice and now in humans as well, where you split the gene into 2 pieces, put 2 halves of the gene in 2 separate viruses, and infect the cochlea with both of those viruses. Otoferlin typically causes profound deafness. It's also identified early on very, very commonly because it has a phenotype, which is auditory neuropathy. So the kids have present otoacoustic emissions and absent auditory brainstem response. And if their imaging shows they have a cochlear nerves, then the odds are pretty good, though not certain, that they have otoferlin deafness. So even before you do the genetics, you have an indicator that this is potentially an otoferlin patient. The cochlear structure is completely normal, at least in mice, at least for a while. In humans, it remains to be seen. And an animal model shows hearing recovery with dual AAV gene therapy. Otoferlin functions at the inner hair cell synapse. So the inner hair cell is normal. The remainder of the cochlea is normal. The spiral ganglion is normal. It's missing, the only thing wrong is that it's missing the otoferlin protein, which drives synaptic release at the inner hair cell spiral ganglion cell synapse. So that's what makes it low-hanging fruit for gene therapy studies, because all you have to do, in principle, is replace the protein. If a bunch of other things go right, it should work. And in fact, it does, not always. So the kids in this trial are identified as having auditory neuropathy. They then get their, and again, they have no ABR and they have normal otoacoustic emissions. And in fact, emissions for the US trials, the presence of emissions are a requirement. In China, they don't care. But in the trials in the US, you have to have emissions. So the patient that I did is a 16-year-old, and she still has emissions. They're not normal, but they're still present. In the mouse model, they undergo cochlear degeneration over time and they lose their emissions. But patients, humans, seem to keep their emissions for extended periods of time. And we don't yet know how viable the treatment is in older humans, but that's part of what we're beginning to find out. As I mentioned, the gene is too big for the adeno-associated virus. It's cut into two. And then this is where the proprietary aspects of the different companies plays a role. They use different ways of getting it to infect inner hair cells. And in the case of the Regeneron trial, they use a myosin 15 promoter. And my understanding is that myosin 15 is only two places in your body. It's in inner hair cells and it's in the spleen. So you're injecting this tiny bit of virus into the scala tympani. And in principle, at least, there's only two places in the body where this gene could get expressed, which is the inner hair cell and the spleen. But again, that's part of gene therapy is the unknown of off-target expression of genes. So how's it delivered? It has to be intracochlear delivery, but that's where the similarities stop. So I don't know about the French company or the Nanjing company, but Refresh Gene in Shanghai and Lilly use a transcanal approach, and they inject it through the round window membrane, and they create a fenestration in the stapes footplate to allow fluid to leave the inner ear. In the case of the Regeneron product, they took my advice, which was it's really hard to do transcanal procedures in 9-month-olds. And all surgeons who do pediatric cochlear implants are pretty versed at opening up the mastoid and the facial recess in a 9-month-old. So I recommended a transmastoid procedure. And instead of stapes footplate fenestration, fenestrating the lateral semicircular canal. That then led to questions of vestibular function, and so that's why we did monkey studies on it, which showed that yes, they take a hit, both hearing in normal monkeys, they take both a vestibular and a hearing hit right after surgery, unsurprisingly, but function recovers over time. So the procedure that the Regeneron folks are using is a very simple procedure for an experienced pediatric cochlear implant surgeon. If this works, I'll show you a quick video of what it looks like. And I'll skip through the stuff that nobody needs to see. This is a 16-year-old with a history of otopharyngeal deafness, received a cochlear implant in India at age 3, developed normal speech and language, moved to the United States, and at age 16, last summer, So here we're fenestrating the lateral semicircular canal. First, we're blue lining it. Same way we actually put in vestibular implants into the canals. You blue line the canal and then you fenestrate it with a pick. And that's our exit point. And in a minute, you'll see the catheter going into the cochlea through the round window membrane. Performed very much like a hearing preservation cochlear implant procedure. And then you turn on this pediatric pump and you can see the fluid coming out of the lateral semicircular canal. That's the fun part of the operation. And then you plug everything with temporalis fascia. This was the first publication. This is from the Shanghai group. This came out in the Lancet last spring. And what it shows is a bunch of deaf ears that have variable recovery of pure tones and ABRs. And by variable, meaning some of them recover close to normal, and some of them don't recover at all, and some of them recover in between. And if you think about all the things that have to work right for this procedure to work, it's not surprising that there's a fair amount of variability. This is the current status of the Regeneron trial. So patient number 1 some of you may know Manahar Vance at Cambridge. Manahar did the first patient in the trial, and that one's a total home run. So that kid got a cochlear implant and gene therapy, I believe, at about 10 months of age, and is now a year and a half later. This is going out to about a year in time, and has nearly normal thresholds. And I'll show you some videos of that child's auditory behavior. Manahar did patient number 2 also, and it had somewhat different result. I hear rumblings that there's some late onset improvement in the outcome in subject number 2, but I don't have the actual number yet, and Regeneron won't tell me. Patient number 3 was done in Spain, and by Manuel Manrique, who some of you may know. And patient number 3 had basically no response at all. And patient number 3 just had a cochlear implant about 3 weeks ago in that ear. So when I say we're in the era where we're doing implants in gene therapy ears that fail, that's the first patient in that group. Subject number 4 is following subject number 1's trajectory, as is subject number 5. Subject number 6 is a 16-year-old, not my 16-year-old, the different one. And subject number 7 is my 16-year-old. So you can see very, even early on, there's highly variable responses from nothing at all to nearly normal hearing. So this is my kid, and this is the audiogram 24 weeks post-op. And clinically, it's really interesting. When she came in, she said, "I didn't know my shower made noise." You think about somebody with a cochlear implant, they take it off to take a shower. They have no idea what the noise of a shower is. Now she can hear her shower. She can't understand speech at all in that year because she's had such a limited response. Her response at about a month or so after this was pretty similar. She's got an ABR she didn't have before. So scientifically, this is still amazing to me. Clinically, it's just not very useful. And we'll see what happens after about a year. And her story is great. You know, she, when she was 3, she saw a cochlear implant surgeon in India, a person I actually know really well. And he tried to convince Dad to do bilateral cochlear implants. And Dad said, "Man, I want to save one ear for future biological therapy if it becomes available." So he implanted one ear. They moved to Seattle in 2015 or something like that, and saw me, and I tried to convince her to get another cochlear implant. Dad said, "No, I want to wait for a biological therapy." And then she volunteered for Ryan Carlson's study of our population, and that's when we found out she had otopharyngeal deafness. So Dad was very pleased with himself. Actually got to do and we'll see what happens over time before deciding what to do next.
So, I have a question.
Yeah, please.
I'm curious, that 16-year-old girl, by the time you saw her, how much of her otoacoustic emissions does she still have?
They were still no, they were still present. You have to have them to be a candidate for the clinical trial, and she still had emissions. They were not normal, but they were present, easily reproducible. And the same is true for the other 16-year-old in the trial. Good question. So this is patient number 1 at about a year. So again, this kid got a cochlear implant in one ear, gene therapy in the other ear. And this is mom testing the child at about a year post-treatment after taking the implant off. So we don't know if the auditory development that you're about to see occurred as a result of the implant or a result of the gene therapy or both, but you can see what the kid can do without their implant on. Mom's covering her mouth too. And here's another example. So obviously, really, let me turn this down. Obviously, really high levels of auditory perception. We can't quantify it very well at this age yet, but it's not subtle what the kid can do with their implant off. So to give you a superficial summary of all of this, there are 5 ongoing clinical trials. Some recipients show dramatic improvements in pure tone hearing months post-op. Speech discrimination is not quantified yet, but clearly it's at high levels, seem extremely likely in some of these patients. So a bunch of questions. What causes treatment failure? What causes mediocre outcomes, like my patient? What will be the outcome of subsequent cochlear implantation on treatment failures? And what about treating an already implanted ear? These are all questions that will need to be answered over time. Another important question is, how long will treatment last? The companies hate it when I say this because there's no publication that says this yet, but there is some evidence that a gene therapy that's FDA approved for a congenital blindness, there's some evidence that it wears off over time and the vision drops. And that's told to me by people that are involved in research on Luxturna. And then the question is, can you redose it? So now they have an immune response to AAV. Now, you've injected it into the cochlea, so how much of a systemic immune response they get is part of what's being tested in this trial. So we don't really know the answer to that. But again, this is something that we'll have to find out over time. Then another big one is cost. Luxturna costs $1 million a dose. Not quite $1 million. They priced it slightly below $1 million just to try and cause less aggravation. And how's this all going to get paid for? In this case of Luxturna, there's no alternative like there is in otopharyngeal deafness. You can get a cochlear implant. So presumably, otopharyngeal gene therapy, assuming it gets FDA approved, will cost less than $1 million and more than $100,000. Somewhere in between those extremes, it's still going to be extremely expensive. And that's why it's so important for us to compare outcomes. And these kids that have an implant in one ear and gene therapy in the other provide a fantastic method of comparing the outcome of the two interventions. And then the last question is, what other genes might be considered? The companies all, of course, want to go after GJP2. But for reasons I'm about to explain, that's a really bad idea. So this is a paper that's actually not in press. It came out in Otology and Neurology a few weeks ago. And Ryan Carlson and Shahar Tiber and I put this together. Shahar is also, and this is the first paper I've ever written with 2 other MD PhDs. Shahar is a resident in Israel. He did his PhD in Karen Abraham's lab and is currently, I think, a PGY2 resident. This is looking at going down the list is the frequency of diagnosis. And the Carlson et al. List is from our center, and Sloan Hagen et al. Is from Richard Smith's lab at Iowa. And it's, the frequency of these different mutations is pretty consistent across the 2. And there are several genes here that have asterisks on them. So TMPRSS3 is the most common gene that has the asterisk. Protocadherin 15 has an asterisk, otopharyngeal has an asterisk, and TMC1 has an asterisk. And what that asterisk means is that by various criteria, it's a good choice to do gene therapy, to develop a gene therapy treatment for. And those criteria are, first of all, how big is the gene? So again, if it's bigger than about 4,800 base pairs, you need dual AAV therapy. So far, triple AAV therapy Wonder why that's.
Someone turned on their microphone. Maybe it's on Zoom.
Can somebody turn off their? Oh, it's gone. No, it's still there. Lianji Zhu. So a good example of a gene that you don't want to chase is STRC. So STRC patients do really well with hearing aids. And if you need to do an intracochlear injection, you're putting them at risk of deafness. There's only one procedure we do where we put an ear that does well with hearing aids at risk of deafness, otherwise known as stapedectomy. Risk of stapedectomy is 1%. So if we can prove that gene therapy can be performed with a 1% risk of ding in the ear, then we can actually consider gene therapy for STRC. I wouldn't recommend we do that until we reach that point. How are we going to do that? Well, the most common gene on this list is TMPRSS3. And it meets all the criteria. In mouse model, it's been shown to respond to AAV treatment, and it produces a predictable hearing loss, as I'll show you in some examples. So DAX was 3. Is there a way to turn the speaker off here?
You could be a believer and fake deafness because one of the old tests was a 30th of a second later play back your own speech. And people can't do it. Your brain allows you to do that. Most people can't do it to do it.
I think I just turned this off. Yes, excellent. So Dax was a 3-year-old boy brought in by his mother in 2008, and she wanted me to do a hybrid cochlear implant on him. And at the time, we were doing hybrid clinical trials in adults. Kids were not allowed in the trial. But this woman, not particularly educated, knew that her son Dax needed a hybrid cochlear implant and knew that I was doing it. I told her that we can't do that because they're restricted to adults. I advised her to wait until FDA approval was obtained in adults and consider off-label use. And the kid went to an oral preschool and got intensive therapy with optimized hearing aids, both at their school and at our CI program. And this was his audiogram at the time. All the things I just told you are the things we knew about him. So a year later, she brings him back, and the therapists and teachers tell her that he's making minimal progress, and mom wants us to put in a regular cochlear implant in the poorer ear and knock off the hearing because she sees the kids in the school with cochlear implants are doing better than Dax was. So I put in a nucleus freedom in 2009 using hybrid surgical technique and a contour advanced electrode via cochleostomy. This shows the postoperative hearing. We lost a little of it. And you can see in the right 6 weeks post-op, he's still got pretty good low frequency thresholds, and high frequency hearing with his implant is terrific. And if you watch over time, 18 months post-op, both ears are declining. The implanted ear is declining faster than the unimplanted ear, but both ears are going down. And he was doing spectacularly well. With both ears plugged at age 6.5, he had 98% PBK words and 100% on child hint sentences. And subsequently, we learned that he had TMPRSS3. And about 1.5 years ago, I did a second side implant, and this year he applied to college, and he really wants to go to MIT. I don't know what the upshot of that is yet. I actually wrote him a letter of recommendation. Really smart guy. This is his. You should know by now. This is his sister, who we implanted a lot sooner because we knew the history, even though we didn't yet know that there was a genetic diagnosis. Interesting, the ears are flipped. The right ear was the worst ear in Dax, and the sister, the left ear, is the worst ear. And in her case, we saved her hearing too, and her hearing remained stable for quite a few years afterwards, but she's subsequently progressed bilaterally. And of course, mom now knows that gene therapy is potentially in the offing, so she's not doing an implant. She's not requesting an implant on the girl's ear, second ear. This is another kid. I met him when he was 6, and he had rapidly progressive sensory neural hearing loss. He went from normal to what you see here in front of my eyes. And I had never seen anything like this in a kid. And I thought, well, rapidly progressive sensory neural hearing loss, Dr. McCabe would put them on long-term high-dose steroids. So that's exactly what I did. And I tortured this poor boy with prednisone for an extended period of time until it was very clear it wasn't doing anything. And we did an implant in the right ear. And this is actually the 6-month post-op audiogram. And parents said, gee, Dr. Rubenstein, you did such a good job of preserving the hearing. Why don't you do the other ear? And I said, let's, he had really good low frequency acoustic sensitivity in the left ear, as you can see. And I said, let's hold off and see what happens. And the family went on a trip to Hawaii, and he had sudden deafness in the implanted ear 6 months post-op. And turns out he has Tempus 3 too. And he's also the youngest person to complete the Seattle to Portland bicycle race. And here he is after finishing the STE. So Tempus 3 is a really interesting disorder because it produces progressive sensory neural hearing loss. Obviously, the ears are very good ears to do hearing preservation cochlear implant surgery on, but they clearly, based on our data now, they clearly all progress over time. Yet early on, they have great low frequency hearing. This shows 5 patients with Tempus 3 and the age at which the cochlear implant occurred and the year in which the cochlear implant occurred and what the progression of the hearing loss is. So that the kid who did the bicycle race is this one. Is the left ear is down to about 60 dB, but we haven't done anything to that ear yet. And as I mentioned, the younger sister of Dax, we haven't done anything to the other ear yet either, because I think this is the next target for gene therapy. Because not only will we be able to potentially restore hearing in these folks, but we will learn what is the risk to hearing, at least to low frequency hearing, of doing gene therapy in ears like this. So it's just like hybrid cochlear implant patients. We learned that we could save hearing with a cochlear implant by implanting people with good low frequency hearing and poor high frequency hearing. And that's who these folks are. I've been talking about phenotypic progression. Here's another type of phenotypic progression. So this is Timmy Brandow. Timmy was the first child in the United States to get a multi-channel cochlear implant. Timmy changed over time. For those of you who don't know, that's Bruce Gantz in 1987 and 2016. Timmy's a biomedical engineer, actually, works in the medical device industry, not in cochlear implants. He's the first kid that I met with a cochlear implant, a first congenitally implanted kid with a congenitally deaf kid with early cochlear implantation that I met. And I was stunned when I met him because he had perfect speech. At the time in Boston, kids were not getting cochlear implants yet. So I'm going to stop there and thank Meg Meredith, who's our senior pediatric audiologist, Mary Claire King, who needs no introduction, Ryan and Shahar Tiber, as well as Regeneron and the Cord clinical trial team. And I'll answer any questions. Oh, and before I do that, I'll try and recruit folks. Anybody interested in cochlear implant technology, that little Gordon conference that had about 35 people at it in 1983 now has 400 people attending, and it's at Lake Tahoe, so it's not far from here. Thank you.